Hello everyone, this is Dr. Adilya. Hope you are all having a great time. Now we are going to discuss about gametogenesis. What is gametogenesis? See, we can split this term simply into gamete plus genesis. Gamete is basically a reproductive cell of an organism, and the genesis means production. So, the production of reproductive cells in an organism is known as gametogenesis. So, we know the gamete in a male is sperm. So, this gametogenesis in male is known as spermatogenesis, and the gamete in a female is ovum. So, this process in a female is known as oogenesis. Now, I need to tell you some basics of genetics before we start into the actual process of gametogenesis. So, generally, all cells in human beings have 46 chromosomes, whereas the gametes alone will have 23 chromosomes. So, this 46 chromosomes is basically represented as 2n or diploid. 23 chromosomes means it is haploid and is represented by a single N. Next, there are two types of process that each cell undergoes. First one is meiosis, where one cell divides into two cells and the number of chromosomes are reduced to half. Okay. So, for example, if I take cell A, it has 46 chromosomes. When it goes meiosis, it forms cell A1 and cell A2, which will have 23 chromosomes each, right? Now, meiosis further has many phases in it. So, this you can remember as PMAT. So, that is prophase, metaphase, anaphase and the telophase. Prophase further has many stages in it, namely leptotene, zygotene, pachytene, diplotene and the stage of dikinesis, right? Then we will have mitosis, where the chromosomal number is constant, that it is maintained. Suppose a cell A becomes cell A1 and A2 to mitosis. If this has 46 chromosomes, cell A1 will also have 46 chromosomes, cell A2 will also have 46 chromosomes. Okay, so this is the basics you have to know. Now we will see oogenesis. The germ cells for oogenesis are the primordial germ cells, and these germ cells are seen in the epiblast by the second week. So, by third week what happens is, these germ cells from the epiblast migrate through the primitive streak and they reach the yolk sac. And by the fourth week, they start migrating from the yolk sac towards the genital ridge, which are the future gonads. And by the fifth week, this migration is complete, right? Now, this is the usual pathway of the germ cells. Suppose if there are any deviation of the pathway of the germ cells, then it can lead to teratoma. Okay, so some of the teratomas include oropharyngeal teratoma and sacrococcygeal teratoma. Teratoma because these primordial germ cells are pluripotent, that is, they are capable of becoming all three germ layers of the body. So we'll see how oogenesis happens now. So there are primordial germ cells. You have oogonium. From that comes primary oocyte. Now this primary oocyte undergoes meiosis 1, but as it undergoes prophase 1 of meiosis 1, it is arrested in the diplotene stage. So there is an arrest at the diplotene stage. This is also known as dictyat stage because here the oocyte goes for a prolonged rest. So can you tell me how long this rest happens? It basically happens till puberty. So what happens during puberty? We all know there is a pulsatile release of gonadotropin releasing hormone where release of FSH and LH happens. Now this LH will help in resuming the meiosis, and then as ovulation happens, just before ovulation, say some 4 to 5 days before, this prophase 1 is completed. Luteinizing hormone will help in resuming the meiosis 1, and this meiosis 1 will be completed 4 to 5 days before the ovulation. Then ovulation occurs where there is formation of a secondary oocyte. At the same time, there is release of first polar body. So what is this first polar body? First polar body consists of most of the cytoplasmic material that is expelled from the secondary oocyte, and secondary oocyte contains mostly a nucleus and some of the cytoplasmic material. Now secondary oocyte now waits till fertilization. Okay, this also enters meiosis 2, but it gets arrested in the metaphase stage. Now this waits till fertilization. Suppose a fertilization happens. So what happens? There is fusion of male and female pronucleus. Just before the fertilization, this second meiosis is completed and there is release of the second polar body, again which contains most of the cytoplasm, and only a female pronucleus and very little cytoplasm is present for the fusion with the male pronucleus. Can you understand? Okay. Now if you see this picture, you will be able to understand it even much clearer. So this is the process of oogenesis, right? So what's happening here is, first there is a oogonium. It undergoes mitosis to form primary oocyte that gets arrested in prophase 1. So this everything happens before birth. Okay, so basically before birth we have a number of primary oocytes. Then what happens is, from during puberty, this primary oocyte just before ovulation completes its meiosis 1 and releases first polar body and secondary oocyte. Now the secondary oocyte enters meiosis 2 and it is arrested in the phase of metaphase till fertilization happens. So just before fertilization, what happens is, it releases the second polar body and it forms a female pronucleus which fuses with the male pronucleus to form a zygote. Okay, so that is the thing. So this is all about the oogenesis in general. Okay, but at the same time I wanted to know about the follicular development also. Okay, so how the follicular development happens? Okay, so first the follicle is known as a primary follicle. See, in this picture you are able to see the primordial follicle first. Okay, I want you to know about the follicular development also. So first one, we have the primordial follicle. So in the primordial follicle, we have the primary oocyte being surrounded by some of the granulosa cells and a basement membrane. Then comes a primary follicle where the primary oocyte is surrounded by basement membrane and a zona pellucida, and the granulosa cells are there. See, what is the difference between here these two follicles is, here the granulosa cells are flat, right? Whereas in a primary follicle, the granulosa cells become cuboidal. Okay, and then in the secondary follicle, there is formation of theca cells. Okay, some of the granulosa cells in the outer region differentiate into theca cells. Then comes the early tertiary follicle or a early antral follicle where there is formation of antrum. Antrum means the space. Okay, can you see the space is being formed, and theca layer differentiates into theca interna and theca externa. Next comes the Graafian follicle. Okay, so this contains the most matured secondary oocyte and the first polar body. And you have the basement membrane, zona pellucida, the granulosa cells surrounding it, theca interna and theca externa layer. See, corona radiata is generally the cells that is surrounding the secondary oocyte, whereas cumulus oophorecus is the layer of granulosa cells that is attaching the secondary oocyte to this layer of Graafian follicles. Okay. So, just beginning of second meiotic division, fertilization happens, and completion of second meiotic division, what happens is just there is a fertilized oocyte, okay, and the second polar body will be released at that time, and surrounding corona radiata will be seen. Okay, so this oocyte will be coming out. So, as the secondary oocyte is released, all the cells surrounding the granulosa cells surrounding the secondary oocyte will be known as corpus luteum. Okay, corpus luteum has two fate. Okay, what if the fertilization happens and what if the fertilization does not happen? If the fertilization does not happen, then it will be known as the corpus luteum of menstruation, where it will be present for 2 weeks, say 14 days, right? So, it will produce the progesterone hormone, and just before menstruation, say some 4 to 5 days prior to menstruation, this will go for degeneration and forms corpus albicans, okay, which is nothing but a scar tissue, white in color. Corpus luteum is generally a yellow colored structure. Okay. Next comes, what if the fertilization happens? Then it will be known as corpus luteum of pregnancy, and in this case, it will be supported by the beta HCG hormone produced by the syncytial trophoblast, and this corpus luteum of pregnancy will be present till 3 to 4 months till the placenta takes over the pregnancy support, right? Now, this is another picture to give idea about the follicular development. So, first there is oogonia. Then we have the primordial follicle, primary follicle, secondary follicle and tertiary follicle, and the pre-ovulatory follicle, which is nothing but the Graafian follicle. Okay. If you ask me, ma'am, how this follicle to be ovulated is selected? It is basically dependent upon the FSH receptors. So, the follicle with the maximum number of FSH receptors becomes the dominant follicle, and from this other follicles which does not have so much of FSH receptors will go for degeneration, and only this follicle persists and it grows. Okay. Now, what is the number of follicles present in a female lifetime? Number of follicles in a female lifetime is maximum at 20 weeks of gestational age, where it is about 7 million, and at birth it becomes about 1 to 2 million, and during puberty only 4 to 5 lakhs are present, the ovulated follicles are very less in number, that is 400 to 600. So, only 400 to 600 follicles are ready for ovulation, and as the number of follicles, one follicle will be ovulated, the rest of hundreds of the follicles will go for degeneration. So, at one point there will be no follicle to get ovulated, and when ovulation ceases, that phase of female reproductive life is known as menopause. Okay, and the fertilizable lifespan of ovum is 24 hours. Okay. Now coming to spermatogenesis. Before going to the actual process, what we have to know is the structure of testicle. Okay. So this is a cross section of testicle where you are able to see the scrotum. The testis is suspended in the scrotum. Okay. We have the tunica albuginea layer surrounding it, and inside you have the seminiferous tubules. Okay. So this is the epididymis. We have the head of the epididymis, body of the epididymis, and tail of the epididymis, which will collectively come into the spermatic cord and coming out through the vas deferens with the nerves and blood vessels. Okay. Now we have interstitial compartment and we have the seminiferous tubules. In the interstitial compartment we have the Leydig cells, then neurovascular bundle. Leydig cells produce testosterone, and in the seminiferous tubules we have Sertoli cells and the germ cells. Okay, Sertoli cells are the supporting cells which form the blood testis barrier. Right? Now, if you take the spermatogonia in the testis, we have two types. One is type A and another one is type B. So, these are mostly the dark spermatogonia and this is the pale spermatogonia. The function of type A spermatogonia is to replenish the stores, whereas this type B spermatogonia alone goes for spermatogenesis. So, it is spermatogonia which undergoes mitosis to form primary spermatocyte, which undergoes meiosis 1 to form secondary spermatocyte. Then from the secondary spermatocyte, we'll have spermatids through meiosis 2, and the spermatid undergoes a special process called spermiogenesis to become a sperm. Right? So, there is no arrest in spermatogenesis as we have seen in oogenesis. And what is this spermiogenesis? Spermiogenesis is a process where the spermatid is converted into a sperm. That is, various structures of the spermatid are converted into a sperm structure. Okay. So, if we see that the nucleus will be forming the head of the sperm, then Golgi apparatus will be forming the acrosomal cap. Then we have the centrioles that is forming the neck and tail. We have the mitochondria that will form the middle piece. Okay. So, the time taken for spermatogenesis is usually 72 to 74 days. Okay. So, this is the time taken for spermatogenesis. But for the sperm to get ejaculated, it has to travel via the ducts, ejaculatory ducts. So for that you may take some additional, it will take some additional 14 days. Okay. And remember, sperm attains maturity in the proximal epididymis and motility in the distal epididymis. The fertilizable lifespan of sperm is about 72 hours. Okay. So this is all about spermatogenesis and oogenesis. Until we meet in the next video, take care and bye bye.